Hello everyone, welcome to week 4. So this week we're just going to go over a brief introduction to cells. We're going to talk about the cell membrane and how different molecules can move across the cell membrane. These are pretty big topics, so I'm just going to really touch the surface on everything, and I'm sure you guys will learn all the details needed in your lecture portion, but I'm just going to touch the surface and give you the information you need for um, the, the lab. So let me go ahead and share my PowerPoint here. Okay, sorry about that. Okay. So the first video is just going to cover activity 1, and then I'll post the next video that will cover the rest of the activities. I think the rest are pretty intertwined topics, so let's go ahead and do this. So the first thing is we have to introduce cells. So all living things are composed of cells, and this is essentially the cell theory. Organisms can be made of either one cell, and this would be referred to as unicellular, or organisms could be made of more than one cell, and this would be referred to as multicellular. So multicellular would be humans, for example, animals, plants, things like that. Unicellular can come from both prokaryotes as well as eukaryotes. So an example of a unicellular prokaryote would be bacteria. And I have an image here. This is Pseudomonas bacteria. So this is an example of a unicellular prokaryote. And this is an example of a unicellular eukaryote. And this is a diatom. So this is kind of these like glass-like algae, and if you've watched the Netflix documentary with Will Smith, I think it's like One Strange Rock or One Strange Planet or something like that. He talks about diatoms. They're really beautiful, and if you want to learn more about them, they're quite incredible. So prokaryotes, they're going to lack a nuclear membrane, they're going to lack chromosomal proteins, and they have no membrane-bound organelles. And here is a depiction of a prokaryote. And this is a depiction of a eukaryote. We're going to go into this in a little bit more detail. So prokaryotic cells, these are found in bacteria, for example. These are very simple and ancient cells. And essentially it's basically think of all the components dumped into a plastic bag. And these cells divide by binary fission. So it's really, I guess, practical for bacteria to be simple and small because it can divide so much more easier. It's really easy for bacteria to divide. It's so simple, so small, and it divides through this binary fission, and it can replicate really rapidly. So if you probably know this, if someone had a bacterial infection in your household and they didn't get it treated quite rapidly, it can spread to other family members pretty quickly. And then you have eukaryotic cells, which are found in plants, animals, fungi, and protists. And these are more complex cells. So some similarities between the two is that they both have plasma membranes, they both have cytosol, they both have chromosomes, and they both have ribosomes. So, and they also both contain DNA. The DNA for a prokaryotic cell is found here in the nucleoid, and then the DNA for a eukaryotic cell is membrane bound in the nucleus. So eukaryotic cells, some more differences to point out are that they have what we refer to as compartmentalization. So these are pretty complex cells, and you have basically certain parts of the cell um, dedicate their structure and both their structure and function to very specific duties. So the best way to think of this is like a home. You have different rooms in your home, and each of those rooms do very different things or serve different purposes. So you have a kitchen, you have a restroom, you have a bedroom, you have like a laundry room. It's at your main bedroom, your kid's bedroom, etc. And that's the same thing here. You have all these organelles, and they have very diverse functions that contribute to the cellular function. Um, and all these organelles are going to be membrane bound, so they're pretty protected. You have a nucleus here, which is membrane bound, and these cells divide through mitosis. This type of cellular division is more complex. There's a lot of checkpoints involved, and it's a longer process, as we will discuss, I think in like a few weeks, I think 2 to 3 weeks. Another thing I want to point out, because if you want to read up on this on your own, it's pretty fascinating, but mitochondria is actually considered to be a one of these simple and ancient cells. It was a prokaryotic cell on its own, and the eukaryotic cell basically engulfed it and then realized, wow, this guy's really useful in eukaryotic cells. Like, there's a huge benefit for having this in our in our cell, and then they ended up just keeping it. So mitochondria were actually prokaryotes. The eukaryotic cell engulfed it and then decided to keep it because they saw benefit to having that structure. And then here are two different eukaryotic cells. This is a plant cell on your left, and this is an animal cell on your right. You are probably pretty familiar with these structures, and you'll probably go in detail over what each organelle's function is. Uh, yeah, but just to point out some similarities and differences here, you'll see that they both have this nucleus, they both have this rough ER, smooth ER, Golgi apparatus, mitochondria. Some differences is you'll see chloroplast here, which is really important for photosynthesis. In the plant cell, you'll see that it has the central vacuole, which is important for water retention. And this is what gives plants, like if you think of plant leaves and their stems, that shape, that turgid shape, that's what contributes to that shape. And then you have a cell wall in plant cells. As you can notice, the cell wall in plant cells is much more rigid and thick compared to the plasma membrane found in animal cells. Or the cell membrane, I'm sorry, the cell membrane found in animal cells. So I'm just going to discuss the cell membrane very briefly. The cell membrane serves several functions. One, it's going to separate the contents of the inside of the cell from the extracellular environment. It is often referred to as a fluid mosaic model because this phospholipid bilayer has many different proteins on it. As you can see here, it has very different proteins. And then in addition, the fluidity of the membrane due to the phospholipid bilayer is where it gets that part of the name from. So as you can see here, our phospholipids, our fatty acid tails are on the inside. We already discussed this in our last class, that these fatty acid tails are comprised of carbon and hydrogen. They are nonpolar, so they're going to interact with each other, and they are considered hydrophobic. The heads are polar. They are composed of a glycerol backbone and a charged phosphate. So these are water-loving, hydrophilic objects. So that's why they tend to put themselves in this configuration. We also talked about the benefit of having a mixture of saturated and unsaturated fatty acid tails. We talked about how if all the tails were saturated, so they were all straight, this cell membrane would be very compact and it would be difficult for molecules to move both inside and out of the cell. So it's good to have some kind of diversity there. So this cell membrane is going to have different proteins embedded in it. So as you can see here, this protein is going to transverse the whole cell membrane and it's going to act as a transporter or receptor, so things can either come in or out. You have some proteins that are just going to be on the top or the bottom or outside or inside of the cell, and you have some that will be kind of like anchor proteins or recognition proteins. So say a molecule is out here floating around and it recognizes this protein, then it could bind to it and it can initiate a reaction inside the cell. So that's really how all these proteins are going to typically work, is that something will either basically come through and have an effect inside the cell through cell signaling, or it could bind and then it'll initiate a change inside the cell, and then there'll be like a signaling cascade that is initiated, which leads to some kind of change in like transcription, for example, or gene expression. So this is once again showing the phospholipid bilayer, how you have your hydrophilic head, hydrophobic tails, how it tends to, how it has this configuration, and how it is embedded with different proteins. So depending on what the cell's function is, these proteins will be very different. So think of a hepatocyte, so a cell in your liver. The proteins a cell in your liver are going to, the proteins a cell in your liver are going to need are very different than the proteins of a different cell, in a different organ or different system. So like brain cells, very different from liver cells. So the proteins embedded in them are going to be very different as well. So this is really dependent on what cell type you're looking at. So you're not always going to have the exact same configuration for each cell membrane in every cell. The other thing is that these cell membranes are semi-permeable. So certain things, typically nonpolar small objects, can just basically come in or leave, just like nothing. Other things are going to need some type of transporter or proteins to help them get in or out of the cell. So it allows some things to come in and move out. So in activity 1, you would requires a microscope, first of all. And here there would be slides that are already prepared for you to view under the microscope. So you would have a slide that's an example of a prokaryotic cell that is already prepared and already put under the microscope. And you would just be asked to simply come up to the microscope, look through the ocular lens or the oculars, and draw what you see. So here you see an example of prokaryotic cells. These are examples of bacteria. So we're not going to go much into it here because this is not a microbiology class, but these cells were stained. It's usually with a certain type of like gram stain, so it would stain gram-negative and gram-positive bacteria. And I actually think we do talk about this further down the line. But essentially, it's going to stain the bacteria either pink or purple. And as you can see here, one bacteria is pink, one bacteria is purple, and that just means whether it has a gram-negative or gram-positive cell membrane or plasma membrane. And then the other thing is you'll notice is a difference in the shape. You'll notice that these kind of have this longer, I guess, rod-like shape, whereas these have a spherical shape. So you would just basically draw this in your lab notebook and write a brief description of what you see. You will also do the same thing but with eukaryotic cells. So there will already be a slide prepared, and you will go look under your microscope and draw an amoeba, which is an example of a eukaryotic cell. So eukaryotic cells are found in the following kingdoms. And when you look under the microscope, you'll see that they have these projections coming off of it. And these are used for its movement, and they are referred to as pseudopods. Um, cells can also have cilia. So actually, I do want to talk about this. So cells can have different types of projections on them. So here you have an example of pseudopods so that the cell can move around. You have other cells that will have projections like such as cilia on them. And that's very common in your example, your throat, like because of the mucus and stuff required there. And then if we go back over here, you'll see that your prokaryotic cells have this flagella, like whip-like projection that will help them move around. But this is not specific to prokaryotic cells. We actually have an example of this in humans. I think the only eukaryotic cell that I can think of that has a flagella would be a sperm cell. So that's an example, or not the only eukaryotic cell, the only um, human cell that has a flagella would be a sperm cell. And cilia is also found in the women's reproductive system. So it's not just specific to the throat, it's just the first thing I thought of. Um, so you would, so those two slides were prepared, the prokaryotic cell and the amoeba microscope, uh, microscopic slides are already prepared for you. You just have to simply see them under the microscope. But you would prepare your slide of this freshwater plant, and to do this, it is referred to as a wet mound. So you would have a microscope slide. So actually these plants would be submerged in fresh water. You would go up to the plant and you would take a leaf off of it with some forceps. So in science, we refer to these as forceps, not tweezers. Take a leaf of it, put it on your microscope slide. Some people will add the drop of water after they place their leaf down. Some people do the water first and then put the leaf on top of that. It's really personal preference. And then you will slowly lower your cover slip onto your microscope slide. So your cover slip's a lot smaller. As you can see, it's a small square. It's still the same glass-like material, but it's smaller and thinner. It breaks very easily also. So you will slowly lower it. I usually use something to guide me down because I don't want to like splash it down. And you also want to avoid bubbles, air bubbles, because it's really annoying under your microscope to get those air bubbles. So you'll put that your cover slips down, and then you're going to remove the excess liquid using a tissue, which we call a Kim wipe in science. So you'll remove that excess water, and then your slide is prepared for viewing. So this is a pretty simple preparation. So you'll take your prepared slide to the microscope, and you will use the microscope as we discussed previously. You'll first turn on your microscope, set up the oculars to your preference, set up the lighting. Remember the general lighting, the condenser, the diaphragm, how much lighting you want to actually hit your slide. And then you're always going to start off with the 4x objective lens and with your stage all the way to the bottom. You put your microscope slide onto your stage. You secure it with the stage clips, and then you look through your ocular lenses, and you start slowly moving your stage up towards your objective lenses. You move it using the coarse adjustment knob, and once you can see it, you start using the fine adjustment knob to focus, and this is all done under 4x. Once you've achieved what you were going for, you move up to the 10x, and there you'll only use your fine adjustment knob, and then once you get achieve what you're looking for there, you move up to your 40x objective lens. Remember, when you move up in magnification, so when you move your objective lens up, you might have to change your lighting settings. But essentially, when you look through the microscope, you're going to see this. These are plant cells, and these are multiple plant cells. So what you see here is the cell wall. And then this is a normal plant cell, so it's healthy, it's nice and hydrated. And then you have your chloroplast here, which are these green ovals. If you play with the lighting, you might be able to see the central vacuole. And then you'll essentially just draw this in your lab notebook and write down the magnification there also. And that's it for activity 1. I'll continue the videos for activity 2, 3, 4, and 5, but they're all pretty similar in topics. So I'll just post one more Zoom recording. The recording that shows the experiments is already up, so if you want to start viewing that, go for it, and then I'll just give you some more context once I get these videos finished. Okay.